Thank you for joining me here in the lovely Winter Park area where I'm coaching the ski team at their Thanksgiving camp. We have been doing a lot of exploration of viruses as a team this week. We have an athlete with influenza and we have another athlete who has the Epstein-Barr virus, better known to all of you as mono or the kissing disease. We've been talking a lot about viruses, and it gives me a wonderful platform from which to talk to you about the history and strategies and morphologies of viruses. Let's start out with looking a little bit at the history of viruses, which we could begin in many, many places. And in fact, it is almost always the case that the first person to discover something is never credited. And there's a wonderful quote by Francis Galton. In science, credit goes to the man who convinces the world, not the man to whom the idea first occurred. And perhaps the irony in that quote is that it quotes simply men as the discoverers. And in fact, it was in the early 1700s when a woman named Lady Wortley Montagu traveled with her husband to Turkey. He was the English ambassador in Turkey. And Lady Wortley Montagu had been very deeply scarred by smallpox. She herself had a very lovely face that had lifelong scars from the disease, but moreover, she had lost her brother to the disease. And so when she was in Turkey, she watched the locals as they actually gave an attenuated inoculum to their children to protect them from smallpox. She tried to come back to England and tell everyone about this, and of course, it was met by deaf ears, and she was unable to make formidable progress in the area of vaccinating against smallpox. It wasn't until the late 1700s, in fact, in 1796, that a man named Edward Jenner, his brilliance came from actually listening to a young woman, a young dairymaid, who had contracted the cowpox and said that that was the reason that she didn't get the smallpox. And perhaps in a dubious encounter, which you can read more about in Baylor's journal of proceedings in 2005, there's an article entitled Edward Jenner and the History of Smallpox and Vaccination. And so in May of 1796, Edward Jenner found a young dairymaid named Sarah Nelms who had a fresh cowpox lesion on her hands and arms. On May 14th of 1796, using the matter from Nelms, her lesions, he inoculated an 8-year-old boy named James. Subsequently, the boy developed a mild fever and discomfort. 9 days after the procedure, he felt cold and lost his appetite, but on the next days, he was much better. And then in July, Jenner inoculated the boy again, this time with smallpox. The ethics here are dubious. The disease developed, but the protection was, or no disease developed and the protection was complete. So this was the first documented, convince the world example of vaccination. So this was the early history, if you will, in short, of viruses. But in the late 1800s, in the 1880s, a man named Charles Chamberlain invented a porcelain filter that allowed him to differentiate viruses from bacteria. And in fact, the term virus is Latin for poison or toxin. And up until the work of Charles Chamberlain, people did not differentiate between infectious agents. So the idea that viruses were a smaller, different kind of infectious agent than bacteria wasn't known until Charles Chamberlain. So he was able to make this filter and see what you all learned in lab, that viruses could pass through the filter, but that, of course, bacteria were caught by and held within the pores of that filter. So the differentiation between viruses and bacteria occurred at that point. In the early 1900s, 1911, there was work done by two Scandinavian scientists, Wilhelm Ellerman and Olaf Beng, very Scandinavian names. They found that there were certain types of cancers, malignancies in chickens that were caused by viruses. And that kicked off a long history of us discovering the link between viruses and cancer. In 1915, bacteriophages were discovered, That is to say, remember that there are viruses that attack bacteria, or AKA germs can get germs, right? So bacteriophages were discovered, and then in 1935, we finally saw the structure of a virus, and that is credited to Wendell Stanley, who crystallized the tobacco mosaic virus, and he was able to show the overarching structure of that virus. So we started to understand this idea that there was a huge difference in the size of viruses as compared to bacteria in most cases. Usually they tend to be 100 to 1,000 times smaller than the cells that they infect. So you can literally see thousands upon thousands of viruses within a single host cell. Some of them are very, very tiny. Parvovirus, if you've heard of that in dogs and wolves, Parvoviridae is a less than 20 nanometer virion. You can think about the fact that we were looking at 1 to 2 micrometer or 1 to 5 micrometer bacterial cells. That is, we're talking 10 to the minus ninth versus 10 to the minus sixth. So hugely different sizes. We would never be able to see a parvovirus on a light microscope. It's far, far too small. You would require an electron microscope. But then that ranges up to the Poxviridae, which are almost starting to get to be the size of some of those tiny archaea that we talked about at approximately 200 nanometers. So you can see that virion size varies greatly, so amongst the world of viruses. Now, when we say virion, we're distinguishing between the extracellular form of the virus, that is the form that occurs outside of the host, versus the intracellular form. So virion is what we call the extracellular form. But what's crazy about that is that when it's in its extracellular form, a virus is pretty much useless. I mean, it sits dormant until it's in a host, but it's only within the host that it replicates. So we call that an obligate intracellular parasite. It requires the host cell in order to replicate. So let's take a look at the differences in virions. Notice that the pox virus here has got a very complex virion morphology, whereas the parvovirus is very regular. It is what we call an icosahedron. So looking at the different kinds of virion morphologies, icosahedral is very, very common. Here in the Winter Park Valley, we were driving from one ski area to the other, and on the side of the road, there's actually a greenhouse that's formed like an icosahedron. Those are some fairly brilliant folks because they know that the most efficient way to enclose space is an icosahedron. 20 equilateral triangles that come together to form this enclosure. In the case of a virus, it's a protein closure, and these are protomers. The protomers are often pentamers that are just regularly repeating structures that encase the nucleic acids of the virus. So if you're thinking about just the nucleic acids inside of the capsid, and the capsid itself, we call that the nucleocapsid, right? The nucleic acids plus the capsid. Now, some viruses are naked, which means that they only have the capsid, the protein capsid, and the internal nucleic acids. There are other viruses that are enveloped, so they're considered not naked. They have an outer envelope, and that envelope may or may not have spikes on it. Naked viruses may or may not have spikes. These spikes are very important in adsorption. In many cases, they have the proteins on them that recognize the receptors on the surface of the host cell. For example, in the influenza virus, the host cell is the respiratory cells within our lungs. So the viral spikes would adsorb to or adhere to those surfaces, those receptors within our lungs. An example of a naked virus is poliovirus, and in that same family is the rhinovirus or the common cold. We also have a morphology called helical, and in this case, the proteins are laid out in a different way. They're laid out as a helix, and they circle up in this helical form, forming an enclosure that is very long and thin. And you'll remember that Ebola is perhaps the most iconic helical virus. It has a very long helical capsid that makes it look like a worm. Tobacco mosaic virus is the one we talked about being crystallized, and tobacco mosaic virus is also helical. Finally, the complex viruses, like we saw the pox virus, but also bacteriophage that we talked about in lab, they're neither purely icosahedral nor are they helical. And there are many viruses that we have come to find fit into that category, maybe particularly because they change over time or there are many variations of their shape. Let's take a look at the different kinds of genomes that we can see within a virus. They are in all types. But one thing that we know, and the way that folks have argued that viruses are not living, is that they don't contain both RNA and DNA. They only have one or the other. So for example, most DNA viruses have double-stranded DNA. So we see our positive sense DNA, our negative sense DNA. This is true of the varicella-zoster virus, or VZV, aka the virus that causes chickenpox. Now I know most of you were vaccinated against chickenpox, but I lived through that and got my long-lasting immunity by having had the chickenpox. Now, RNA viruses also abound, and most RNA viruses are single-stranded RNA. So they may be positive sense RNA viruses, or they may be negative sense RNA viruses, but most of the time they're single-stranded RNA viruses. A good example is the rhinovirus. It is a single-stranded RNA virus, but there are many others as well. And influenza, while it's a single-stranded RNA virus, is something that we call segmented. That means it has greater than one strand of RNA. Influenza has 7 or 8 strands of single-stranded RNA. There's also double-stranded RNA viruses. These are far less common, but the rotavirus, we've pretty much all lived through that. If you got a case of tummy upset, gastroenteritis, pretty much everyone has had a rotavirus. Rotaviruses are actually double-stranded RNA. In fact, at last count, there's hundreds of thousands of children worldwide and annually, actually greater than 800,000, that die of rotaviruses because they die of dehydration and diarrhea. There's also single-stranded DNA viruses. This is, for example, our parvovirus that we saw on the prior page that we've talked about particularly in dogs. Now, there's also human parvoviruses. There's one called the B19 that can cause disease in humans as well. So either DNA or RNA, either single-stranded or double-stranded, but they're not both. They don't have both. And that's why for many years that has been argued as the case for them being not living. We know that there have been now some new arguments that they should be categorized as living. And some of those come from the conservation of protein folds. Let's take a look at your pre-party for today. For your pre-party, this is going to take a little bit of evolution, and I want you to start working on it right now. You may have to continue to work on it through our next class, but I want you to sit down and do a little bit of writing and a little bit of drawing. And I want you to pretend that you are poliovirus. Now, poliovirus, it's one of those viruses that folks like to say, woohoo, we've eradicated poliovirus, but it's just not the case. And I tend to think of eradication in that term as sort of being a curse word, because I think we need to bring balance to all forms of being within our Earth, as opposed to trying to eradicate certain beings, whether they are living or not living. So here's our story. Your virion is naked and icosahedral. You're very picky about your target cells. You recognize a receptor on the surface of human, nasal, pharyngeal, and neural cells. Your genome is positive sense RNA. Okay, think about what that means. This RNA is recognized by host translation machinery. Interesting. Okay. All right. Sounds like you've got some direct strategies going on. Your replicative intracellular form is double-stranded RNA. However, you like to keep this at low concentration because it's antigenic. Please draw the things that must occur to allow your genome to get into the cell, replicate yourself, encode for ample protein, and exit your host cell so that you can invade another. And we're both going to draw this and write about this and write a narrative about this journey that you take. So begin to think about that. And I can't wait to see you in class because we're going to be playing a rousing virus game.